next speaker is David Schneider from Stanford University, talking about defining a new alphabet. Describe host trade-offs turned infections.
Yeah, I think when I made that title, I was feeling kind of panicked when I gave it to Marty. Maybe in 5 years, this is where I hope we'll be, but I'll tell you what we're hoping to do to head us in this direction. So I work in mostly on insects. We look at resistance and tolerance to infections, and I work at a medical school where most of the people I work with think that the role of the immune system is to cause rejection of organs and autoimmunity. It has little to do with infection. And though I work on insects, I take care to carefully isogenize everything before I start and get rid of any natural variation. So I'm kind of stuck between two worlds here. But we've become very interested in resistance and tolerance, and I'll talk about how we think about things, and I'll try to be careful about including Lars' work and Andrea's work and Tom's work, because I tend to simplify things a little bit, and I want to make sure that I'm not picking fights with them, but I agree. I just have to simplify for some of the things that we're able to do. So the reason we got into this is from a genetic screen that we were doing. We were taking flies, we were infecting them with a completely artificial pathogen, with Listeria monocytogenes, and we looked for flies that died rapidly. And we found a dozen strains where the flies died really quickly and the bacteria grew out of control. And we knew what to do with these. This is what we expected. These were resistance mutants that were unable to control the infection. The bacteria grew like crazy. But we found a third of the mutants were just as able to control bacterial growth as were wild-type flies, but the flies dropped dead. And this was, I think, we'd actually encountered these before, but not systematically and not at such a high concentration. So we realized that, roughly a third to half the time we made mutants in flies, we ended up with flies that couldn't fight the infection. And they couldn't fight it because they just didn't survive the infection. We had to start thinking about ways of discussing this. And so the question is, how can you summarize an infection? So here's what happens in an infection. Take this guy, let's say hello, infect him, what the heck, and then he gets infected and he gets better. And the question is, how do you summarize this? What two data points do you take out of this so that you can then talk about it and compare? So what you can do is you can take a timeline where you look at split it into frames, take the number of microbes you've got, take the amount of health you've got over time, and look at, say, maximum microbe number and minimum health, and plot these. And this is, through most of the talk, I'm actually not using my own data. I'm using some data from work that was done here in Andrew Reed's lab by this graduate student. And this is nice because they got this data set with, I think it's 58 mice that are infected with Plasmodium chabauti and monitored parasite density and red blood cell counts. And you can do a lot of neat stuff with this, and this is what I'm going to play with mostly today. So what she did was infect mice at concentrations varying from 100 to 10 to the 8th parasites. You'd see you get different peaks. The parasitemia would come up at different times depending on what dose you gave them, and you'd end up with different minimal red blood cell counts. So you can take this data and process it and make a really beautiful tolerance curve. And I just wanted to show you this because it's the regularity of this is wonderful. When you plot the data, it comes up like this. This is done with 2 strains of parasite. I think the CW strain is from blue to green, and then BC is this dark green to red. And granted, you could probably plot 2 different curves through this if you wanted to. You could plot this one curve through this parasite and then a really flat curve through the second one. But if you put them together, you get something like this. Okay. So, and when I talk about this, and I'm following what I think Lars had started, is that I step away from using fitness as an output and start to use health as an output. And when you do this, you can it gives you a lot of freedom and it creates a lot of trouble. The nice thing about fitness is there are a relatively small number of things that you have to look at. When you look at health, you can say anything you want to as health. Anything that you're interested in can be health. And that becomes interesting because you can then start making enormous curves where you're comparing millions or thousands of factors to each other, and we'll get towards that towards the end. Okay. So in practice, it's really hard to get a beautiful tolerance curve like this. That's a spectacular amount of data. when we do these experiments, what we can mostly generate is this, okay? I can find vigor pretty easily. For me, I look at the health index of the animals before I've infected them. And I can get a really good point out here where I have a lot of data at 1 microbe number to figure what the health index is. And so what we've been doing in our studies is trying to figure out what happens to this point when we change the conditions, when this point remains rather constant. And so what we ended up doing was asking, now I know a little more math, so I can describe this more mathematically. I understand what I was doing. I was thinking about it as if we took the equation and linearized it. So we're looking at this at such a magnification that the area around the point we're interested in is basically linear. And we're asking what happens when you change the conditions. So you can imagine two things happening. If you change just the resistance, your point should slide up and down this tolerance curve. If you're changing just tolerance, it should go up and down. But if resistance isn't changing, you'll end up with these two things happening, and they could be independent of each other. And if both of these things are happening, what we imagine when we change conditions is that you'll end up with this two-dimensional health by pathogen load space. And we can make guesses about what's happening in the system when this point moves. Okay, so this point could either stay in this one place or move to the basic 8 different directions of the compass. If it moves straight up or down, we'd say that's a change just in tolerance. If it's sliding up and down on the diagonal, we'd say that's a change in resistance. And if it's moving to the left or right, that has to be balanced changes in resistance and tolerance, where one goes up and one goes down. And so we've looked at what happens in a number of different flies under different mutant conditions, or flies where we've altered the environmental conditions, and then hit them with a number of different pathogens. For example, we see that when we take flies and knock out one of the immune responses, where the fly makes melanin, makes reactive oxygen to kill a parasite. And then we infect the fly with 6 different pathogens, we get 5 different results. And this was really exciting because it was supposed to be a control experiment for a paper. And what would happen, my student Janelle, when she'd come in each day and say, oh, I got the results, and then she'd try another pathogen, the next day she'd say, I got a different result. And for about 2 weeks, she was coming in with these different answers. And when she ended up getting 4 results that sent it in different directions, we realized we could write a whole paper on this control. And so what she saw was, you infect these mutant flies, they're lacking this immune response. With E. coli, there's no change in the infection. Okay, so I should point out, vigor never changes in these. The intercept here is always the same. So E. coli, there's no change. If you infect with Listeria or Salmonella, you see an increased number of bacteria, you see a decreased time to death. We think this is a movement just down the tolerance curve like this. With Streptococcus pneumoniae, we see this odd result where we remove the immune response and the fly becomes better at killing the microbe. And then we see some situations like this here. If we infect the fly with Burkholderia, the fly becomes better able to survive the infection, but it has more microbes in it. We see the reverse here, where we infect with E. faecalis, it's better able to clear, but presumably doesn't do as well. But that's at least our assumption, because we see changes in microbe level where health remains constant. Okay? So if you're changing the microbe level, you've got to have a change in tolerance in this situation. And so we're kind of in Hershey heaven here. We've done this again and again and again, and every time we do this with different environmental conditions or with different mutants, we see the same general sort of effect, that every time we do this, you see at least 3 different possibilities, up to 5 different possibilities. So the point here is that often theoretically when we talk about optimization and what happens in the system, we're talking about what happens when you're putting one parasite in a host. And Paul pointed out that that's not what happens in the wild, and what this is showing is that when you change the system, when you change the host, it alters the way you get an interaction with every microorganism, and just changing one mechanism can result in changes that affect either resistance or tolerance. So it makes me think that there aren't just resistance or tolerance genes, that these are system level effects, and we have to talk about them as system level effects. Okay. So what causes tolerance? And so we can talk about this in a couple of different ways. What I'd like to be able to do is to be able to study, Donald Rumsfeld called these the unknown unknowns. Okay, I want to be able not to just study things that I know about, say, go and look at a toll signaling pathway and ask what will cause tolerance here. I want to be able to go into the system and say, I want to be able to find things that alter the slope of this health by microbe curve, even if I don't know what the mechanism is. So I've come up with a set of sort of anthropomorphic, or you'll see Davidomorphic versions of this. And I could have gone mathematically, but then I end up talking about ratios of different things, and I think it's kind of opaque. So I'm going to go with the sort of anthropomorphic version first. So the thing that's easiest to pick up when you're talking about tolerance is that you have to deal with damage. And there are basically three different ways of dealing with damage. Everyone talks about the double-edged sword. I don't think that's really a great metaphor, because the way the sword is designed, it's actually that you've got a handle, and then it's got a pointy end, and you point the pointy end away from you. Okay, so and that's a tolerance mechanism. That when you hold it, it's not cutting you. Okay, so tolerance that you work with when immunologists think about tolerance, that you're not making antibodies or specific reactive T cells, that's a type of tolerance. And it's a tolerance where you're making sure that the pointy end is pointed somewhere else, that you aren't making things that are going to be self-reactive. Over evolutionary time, presumably, we've evolved antimicrobial peptides so they don't cause self-harm. It's another example of putting the pointy end somewhere else. Okay. So as I was writing something on this, I started thinking about, well, what else does a knight need when they've got this weapon? What do they need to avoid hurting themselves? And a friend pointed out that the knight doesn't walk around naked. Okay, so the knight is wearing armor not just so that they will not get cut by their own sword, by someone else's sword, but their own sword. So armament is going to end up being a kind of tolerance to prevent self-harm. And of course, if you end up being injured, you have to repair that as well. And that's going to show up as a tolerance effect too. I think efficiency of immune responses show up as tolerance as well. So one class, and this is a cartoon. So this is overindulgence. So if you have an immune response that produces a bigger reaction than it needs to, it will cause damage, it will use too much energy, and I think of this as an efficiency mechanism. There are also efficiency mechanisms that I consider ignorance. This is not seeing a microbe that's there. For example, the way you might ignore guts in your intestine, or maybe the best example that's come out recently was with the sequencing of the aphid, where it was found that these lack the entire IMD signaling pathway. And the idea that Angela Douglas put forward on this is that they might lack this pathway because they have essential endosymbionts that would be recognized by this pathway. And one of the ways they deal with it is by ignoring it. Okay, that would be a passive ignorance, where they just don't get the genes to do it. There's also active ignorance, where you could say have mucus that prevents microbes from getting close enough to your epithelial wall to be able to recognize them in the gut. Okay. And then I think a similar sort of thing would be silence. A good example of silence would be immunoprivilege, where you could actually detect something in a tissue, but you are designed not to detect something in that tissue because where you can detect it, but you're not going to respond to it.